Welcome everyone. This video introduces our research on solving the EEG inverse problem using deep learning methods for enhanced brain activity localization. Our work proposes a lightweight vision transformer architecture that improves localization accuracy while significantly reducing computational requirements. First, let us understand the clinical motivation behind this research and the problem we aim to solve. Epilepsy is a neurological disorder characterized by recurring seizures caused by abnormal electrical activity within the brain. Patients may experience symptoms such as involuntary muscle movements, temporary loss of consciousness, confusion, or changes in sensory perception. Accurate localization of the seizure origin is essential for effective treatment, especially in drug-resistant epilepsy where surgical intervention may be required. Today, clinicians mainly rely on EEG recordings and MRI imaging. However, identifying the precise source of abnormal brain activity remains a challenging and time-consuming process. Electroencephalography, or EEG, records the brain's electrical activity using electrodes placed on the scalp. It is completely non-invasive, inexpensive, and provides excellent temporal resolution, allowing clinicians to observe brain activity in real time. These advantages make EEG one of the most widely used tools for neurological diagnosis and continuous brain monitoring. To understand our research, it is important to distinguish between the forward and inverse EEG problems. The forward problem predicts scalp EEG measurements when the brain source locations are already known. The inverse problem works in the opposite direction. Given only the EEG recordings, it estimates where the underlying brain activity originated. Successfully solving this inverse problem enables applications including epilepsy diagnosis, cognitive neuroscience, brain-computer interfaces, and neurofeedback systems. EEG source imaging has evolved considerably over the past century. The field began with the invention of EEG in 1924, followed by dipole modeling, mathematical forward modeling techniques, and inverse algorithms such as Loretta and S-Loretta. More recently, advances in artificial intelligence and deep learning have enabled data-driven approaches capable of learning complex mappings directly from EEG signals. Despite these advances, several important challenges remain. First, the EEG inverse problem is mathematically ill-posed, meaning multiple brain source configurations can produce nearly identical EEG measurements. Second, many existing deep learning models require extremely large synthetic datasets, often several terabytes in size, together with weeks or months of simulation time. Finally, these computational limitations prevent many methods from becoming practical clinical tools. Therefore, there is a clear need for a lightweight, scalable, and clinically applicable solution. Our research addresses these limitations through three objectives. First, we develop a lightweight deep learning model that achieves high localization accuracy using significantly smaller training datasets. Second, we introduce a novel vision transformer architecture capable of simultaneously learning spatial and temporal EEG features. Finally, we validate the proposed model using clinically relevant EEG recordings to demonstrate its practical applicability. Before introducing our solution, let us briefly review the most influential deep learning approaches for EEG source imaging. ConvDip was one of the earliest convolutional neural network models developed for EEG source localization. It converts EEG measurements into low-resolution two-dimensional images and processes them using a shallow CNN. An important design choice was avoiding pooling layers, allowing the model to preserve fine spatial information required for accurate localization. Although pioneering, ConvDip only processes individual EEG snapshots and does not capture temporal dynamics. When evaluated against traditional algorithms such as E-Loretta, LCMV beamformer, and CMEM, ConvDip demonstrated improved localization performance. However, its inability to model temporal relationships limits its effectiveness for realistic EEG sequences. DeepSIF represents a significant advancement by introducing spatiotemporal learning. It combines a ResNet-based spatial module with an LSTM temporal module to reconstruct evolving brain activity over time. This enables more realistic modeling of dynamic neural processes compared with purely spatial CNN-based approaches. DeepSIF introduced several important innovations, including biologically realistic training data generated using neural mass models. However, these improvements come at the cost of substantial computational complexity. Generating the required training data demands extensive storage and simulation time, while the model itself contains millions of parameters, limiting scalability. This comparison summarizes the strengths and weaknesses of current approaches. Although deep learning models outperform traditional mathematical methods in many situations, their performance often decreases when tested on unseen datasets. This motivated the development of a more generalizable and computationally efficient architecture. We now move to the proposed methodology of our research project. Two complementary simulation frameworks were used to generate training data. The first is SEREEGA, which efficiently produces synthetic EEG recordings with precisely controlled source locations. The second is neural mass models, which simulate biologically realistic neural activity using the Janssen-RIT model. Using both datasets allows the proposed model to learn from diverse and representative EEG patterns. Our proposed model is called ViTESI, a vision transformer designed specifically for EEG source imaging. Instead of treating EEG as images, we represent each time step as an individual token. These tokens are projected into a shared embedding space, and temporal positional encoding preserves the sequential order of the EEG signals. This formulation enables transformer-based learning while maintaining the temporal structure of EEG recordings. The transformer encoder applies multi-head self-attention to capture long-range relationships across the complete EEG sequence. Unlike recurrent networks, all time steps are processed simultaneously, improving computational efficiency while learning both temporal dependencies and inter-channel interactions. Finally, the encoded representations are mapped into spatio-temporal brain source activity. To assess clinical relevance, we evaluated the model using two independent real EEG datasets. The first contains recordings from patients with drug-resistant focal epilepsy, together with clinically verified seizure locations. The second is the publicly available visual evoked potential dataset, where activation is expected in the primary visual cortex. These datasets provide realistic benchmarks beyond synthetic simulations. We now present the experimental evaluation of the proposed model. When trained and tested on the same dataset distribution, ViTESI consistently achieved the best or near best performance across localization accuracy, reconstruction quality, and temporal consistency. These results demonstrate that the proposed transformer architecture effectively learns complex EEG source mappings using relatively small training datasets. We next evaluated generalization by training and testing on different simulated datasets. Even under these more challenging conditions, ViTESI maintained superior performance across almost every evaluation metric. This indicates stronger robustness and improved generalization compared with existing deep learning models. We also investigated the effect of training dataset size. Performance improved rapidly as more training samples were introduced. However, beyond approximately 6,000 samples, further improvements became minimal. This suggests that ViTESI can achieve competitive performance without requiring extremely large datasets. Qualitative comparisons show that ViTESI produces localized brain activity patterns that closely resemble the ground truth. Compared with existing approaches, the predicted sources are more spatially focused and contain fewer false activations. When evaluated on real epilepsy patients, ViTESI produced source estimates that aligned closely with clinically identified surgical resection regions. These findings demonstrate the potential of the proposed model to support clinical decision-making in epilepsy treatment. On the visual evoked potential dataset, ViTESI successfully localized activity within Brodmann areas 17 and 18, which are responsible for visual processing. This confirms that the model generalizes well to different types of real EEG recordings. Besides achieving strong localization performance, ViTESI is also computationally efficient. Compared with DeepSIF, it requires significantly fewer trainable parameters and considerably fewer floating point operations. This improved efficiency makes the model more suitable for practical deployment and future clinical applications. To demonstrate practical usability, we developed a web-based visualization platform. Users can upload EEG recordings, perform source localization, and interactively explore predicted brain activity across different time points. This prototype illustrates how the proposed research can be translated into a usable clinical decision support system. Before concluding, we will now present a short demonstration of our EEG source visualization tool. Here, the user can upload an EEG signal file directly into the system. Once uploaded, the server pre-processes the signal and runs it through our trained ViTESI model. The predicted active brain regions are then displayed on an interactive three-dimensional brain model. Initially, the system shows the peak activation time point. Users can also adjust the timeline to observe how brain activation changes over time. Thank you for watching. This project demonstrates how lightweight transformer-based deep learning can make EEG source localization more accurate, efficient, and closer to real clinical use. We hope this work contributes toward faster and more reliable brain activity localization in future neurological diagnosis.